I am Paulo Henrique, Commission Editor at River Publishers, and today I have the great pleasure of welcoming Professor Andrea Tangherloni from Bocconi University in Italy. Professor Tangherloni is a leading researcher at the intersection of computational science, biology, and medicine, with a strong track record in data-driven biomedical research and translational computational methodologies. This episode inaugurates an exciting new initiative at River Publishers, the Rapid Book Series Computational Approaches in Biology and Medicine. Professor Tangherloni, it's a pleasure to have you with us today. Welcome.
Thank you so much, Paolo, for the great introduction. I'm Andrea Tangherloni. I'm an assistant professor at Bocconi University in computational biology and AI. Basically, I try to combine AI approaches with computational biology to tackle complex problems in biology and medicine. I did a bachelor degree, a master degree, and a PhD degree in computer science at the University of Milano Bicocca. And then I did a postdoc in Cambridge. And then I did another postdoc here in Italy. And since 2023, I'm an assistant professor.
The first question to you is related to the motivation, the journey of this series. Professor Andrea, the launch of this Rapid series is a significant achievement. Could you share with us the journey that led to its creation? And also, what motivated you to initiate the computational approach in biology and medicine series? And what gap did you feel it was important to address?
Oh, well, if I really look back, the roots of this series go all the way back to my master thesis and continue through my PhD. I was fortunate to have supervisors who followed me across both stages and were extremely rigorous. They constantly emphasized the importance of scientific method, of being explicit about assumptions, and of ensuring that results could, at least in principle, be reproduced by anyone else. At that time, it felt demanding, I can confirm, but later realized how formative that training was, and it shaped the way I think about research of this day. As I moved into biomedical research during my master thesis and my PhD, I began to notice a recurring problem. Many papers published in high-impact journals contained serious methodological flaws, such as statistical issues, questionable machine learning practices, and unclear experimental design. Even with access to the data, in many cases, it was simply impossible to reproduce the results. That was frustrating, but also motivating at the same time. And the series was born from the idea that we need resources that explain how to do biomedical data analysis properly, not just which tools to be used, but also how to reason scientifically, validate the achieved results, and make analysis reproducible. This is what I wanted this series to stand for. During my PhD, I also spent periods abroad in both the United States and the UK. And these experiences were eye-opening for me. I saw different research cultures, different expectations. I worked in an experimental lab, and I was essentially the only person with a very strong computational background. That position came with a responsibility, I would say. I pushed very hard for all analysis to be fully documented, for the code to be readable and version controlled, and for the data, script, and also the results to be made available for everyone in a public repository, obviously along with the published paper. It was not an easy task, but the outcome was extremely rewarding, I would say, because the results we published in a high-impact journal are completely reproducible. Anyone can take the data, follow our analysis, run the code, and obtain exactly the same figures, conclusions, and results. That experience convinced me that rigor and openness are not obstacles to impactful research, but they are what make impact sustainable. And this is very crucial and important for me. Now, as an assistant professor, I explicitly share this philosophy with my students. I tell them that results are not enough. Their approach must be rigorous, their core understandable to others, and their analysis reproducible. If results can be explained or rerun, that it's not finished science. This series is really a formalization of that trajectory, I would say, from training to international experience to dependent research and teaching. It's an attempt to distill these lessons into concrete practical guidance for biomedical researchers who wanted to think properly.
Now, talking about this series in terms of the editorial team that you assemble, as editor-in-chief, you have assembled a strong and highly respected editorial team. Could you tell us how you identified and brought together these experts?
So, although I am the editor-in-chief, this is very much a collective effort, I would say. When selecting the editorial team, I focus on people who combine three qualities: deep technical expertise, hands-on experience with real biomedical data, and a strong commitment to methodological rigor. I wasn't interested in assembling a team based solely on prestige, I would say, but I wanted the contributors who can care about how results are obtained, not just where they are published. Many of them are people I'm collaborating with, reviewed alongside, or whose work I have followed closely over the past 5 years, I would say. Yes, I do plan to extend the team as the series grows into new domains, for instance, multi-omic integration, foundational models, generative AI. We will need additional expertise and leading people in that area, in those areas. Expansion will be carefully and I would say purposeful, not just to increase volume, but to maintain a coherence and the quality of the series, because the quality is much more important than I would say the numbers.
Series. Could you outline the overall structure of the series and how many volumes are planned and what the thematic areas you intend to cover?
So I would say, myself and the team designed this series as a modular but coherent ecosystem. Each book is self-contained. You can read one without having read the others, but they are also conceptually connected. We start with foundation volumes that establish shared language and methodology. From there, we will move into more specialized topics. Reader can follow the full trajectory or jump directly to the domain they are care about most. But the structure reflects how real biomedical research works. You need strong foundations, fundamentals, but you also need depth in specific technology and data types. So there will be at least 3 more books, and then we will try to extend the series with new approaches and new data, because also the biological part, it's growing a lot with new technologies, and so we will able to produce new data, and we
That's
will
good.
need more computational frameworks to analyze them.
So also I know the first volume, Biomedical Data Science, a step-by-step guide to analysis and interpretation, is now underway. Could you give us an overview of its key contributions and explain how it sets the conceptual and methodological foundation for the future books in the series?
Oh yes, our first book is intentionally foundational. It focuses on experimental design, statistics, machine learning, and reproducibility, not superficial, but not at a superficial level, but in ways that are directly applicable to biomedical problems in real life. A key feature is that we don't just explain methods, we explain why and when to use them, and just as importantly, when not to, because it's very important. We also integrate the executable code and reproducible workflows, because methodology without reproducibility is incomplete.
That's really good. From your perspective, Professor Tangherloni, what impact do you expect this series to have on biomedical research, particularly in terms of data interpretation, methodological rigor, and the translation of computational insights into clinical and biological practices?
So, my hope is that the series helps shift the focus from state-of-the-art all cost to sound, transparent, and reproducible science. If readers come away with a better understanding of statistical assumptions, also, I would say a healthier skepticism towards overly optimistic machine learning results, and the habit of properly validating and documenting their analysis code, then the series has succeeded, because basically I would say that ultimately better methodology leads to better biology, and better biology leads to better clinical translation. That's the real impact I care about more for this series.
Looking ahead from now, how do you plan to expand the series? What emerging areas or topics do you anticipate exploring in the upcoming volumes? And for instance, how do you see the series evolving in response to advances in AI, computational biology, digital health?
So to be honest, we have already started working on two domain-specific books. One on RNA single cell analysis, but also RNA bulk analysis, and one on attack sequencing data analysis. These go deep into the practical and methodological changes of those technologies and modalities, while remaining grounded in the principles established in the first volume. Looking ahead, one of the most exciting direction is multi-omics integration, especially in the context of recent AI developments. We are exploring how modern machine learning and deep learning models, such as representation learning, foundational models, generative approaches, can be applied to integrate heterogeneous biological data with reproducibility and also responsibility. Importantly, we are not chasing trends. Every new volume will be driven by the same question. Does this help researchers to do better, more real-world science or not? This is the most important question we try to answer with this series.